Hi, everyone. Thanks for coming. Today, we're going to talk about one of my favorite topic, which by the end of this talk might be one of your favorite topics. And that's about fibroblasts, which are one of the cell types that are part of the tumor microenvironment in solid malignancies, such as pancreatic cancer. So why do we focus on pancreatic cancer? Well, that's because we have done, and in the past decades, there has been quite a significant improvements in patient survival for a number of malignancies. And that's because we have made progress in diagnostic tools and therapies for many cancer types. Unfortunately, this is not true for pancreatic cancer, for which the patient survival rate really remains very low. And while this is a graph that is up to 2011, the column for this year, a decade later, wouldn't be much different. So why is this malignancy so lethal and with such poor prognosis? Well, there's a number of reasons. The predominant reason, perhaps, is that this is a disease that is diagnosed at very late stage when it's already spread at metastatic sites. And so for these patients, surgery and removal of the primary tumor is not an option. Why is pancreatic cancer diagnosed so late? Well, also for that, there's a number of reasons. It's in a location that's not so easily accessible, like for other cancer types, such as melanoma and breast cancer. There's no screening that is ongoing, such as colonoscopies and mammographies that have improved the survival and the early detection for other cancer types. There's no biomarkers that we can look at to evaluate whether a patient has pancreatic cancer or not. But also, even if those diagnostic tools existed, because pancreatic cancer is overall a rare malignancy compared to many other cancer types, it wouldn't be feasible to screen the population with these diagnostic tools because we would end up having really high clinical costs and also several false positives. So while the early detection of pancreatic cancer is necessary and much needed, we also need new therapies. And that's because we actually have very limited treatment options for patients. And when I say that the reason why poor survival is linked to the fact that this disease is diagnosed when it's metastatic is because to date, the only curative option for pancreatic cancer patients is surgery. And patients that present with a metastatic disease don't have several options. There are two main regimens, one called gemabraxane and one called FOLFIRINOX. With FOLFIRINOX, that is perhaps more effective, but it's also harsher, and so it cannot be given to every patient, especially considering that the majority of pancreatic cancer patients are elderly people, and so they are more frail and cannot receive this type of treatments. So we need better therapies, but there's an additional layer of complexity of pancreatic cancer, and that is more chemotherapy resistant than other malignancies. And one of the components that contributes to this chemotherapy resistance is the tumor microenvironment. And so it's clear that to identify new diagnostic tools and new therapies for this disease, we need to understand better the biology of this malignancy. And it's emerging now that just by looking at the biology of the cancer cells, understanding the genetic and the epigenetic changes that occur in the cancer cells of pancreatic tumors, we only have a very limited picture of the biology of these tumors. And the reason is that the majority of the components of pancreatic tumors are actually non-cancerous cells, which can comprise up to 90% of the overall tumor mass. And so paradoxically, you can think that one of this lethal cancer has as little as 10% of its malignant mutated epithelial component, and the majority is comprised of these non-cancerous cells. So in my lab, we are interested in understanding the biology of these non-cancerous cells, how they interact with pancreatic cancer cells, and how we can stop these tumor-promoting interactions. And among non-cancerous cells, we are particularly interested in cancer-associated fibroblasts, or CAFs, which are going to be the topic of this talk. And we want to understand how these normal fibroblasts are hijacked by the cancer cells and what roles they play in the tumor microenvironment and how we can target them. And while we will go through the several reasons and roles that these CAFs play, one of the most obvious reasons of why someone should be interested in fibroblasts in pancreatic tumors is that they are the most abundant cell population in the tumor microenvironment. And that they are also the population that is largely responsible for the deposition of the extracellular matrix, which you can see here as this blue ocean of collagen that surrounds and embeds the little islands of cancer cells. But where are CAFs coming from? Well, in the lab, we work predominantly with fibroblasts, resident fibroblasts, and pancreatic stellate cells, which are a type of a specialized type of fibroblast. And that's because we are a bit conservative with the definition of cancer-associated fibroblasts. But there has many studies that have shown that other cell population can acquire CAF feature in the context of tumors, and not just in pancreatic tumors, but also in other malignancies. These are predominantly in vitro studies, and more lineage tracing studies will be needed to really make sure and validate these results. But you may find on this slide your favorite cell type and see that epithelial cells, mesothelial cells, adipocyte, endothelial cells, pericytes, and cells recruited from the bone marrow, they can all acquire CAF features in the context of a tumor. And how do CAFs become CAFs? Well, again, in the lab, we are predominantly interested in understanding how epithelial cues are reprogramming normal fibroblasts or pancreatic stellate cells to become CAFs in this activated state. But there's a number of other factors that include stromal cues from other immune cell types or from other CAF subtypes that can affect surrounding normal cell types. Oxidative stress is also involved in this reprogramming, and so is metabolic reprogramming, regulation through microRNAs, and epigenetic changes, which we are also interested in investigating. And what roles CAFs play in the tumor? Several roles, and this is true not only in pancreatic tumors, and in fact some of these references are for colon cancer and breast cancer. They, as I mentioned, are responsible for the deposition of the extracellular matrix, which in itself plays several roles. It can provide biochemical and mechanical cues that activate a number of tumor-promoting signaling pathways in the cancer cells. But ECM proteins have also been shown to act as nutrients. They're taken by the cancer cells and fuel their growth. And also ECM components have been shown to act as a barrier to drug delivery, and so increasing chemotherapy resistance. CAFs also contribute actively to chemotherapy resistance, for example, by scavenging gemcitabine, which is the standard of care of pancreatic cancer, and so lowering the overall concentration of the drug within the tumor and leading this to affect less the cancer cells. CAFs also secrete a number of ligands, which promote the proliferation of cancer cells, metastasis formation, and also the establishment of an immunosuppressive microenvironment. And as I mentioned previously, chemotherapy resistance and metastasis formation are key features of pancreatic tumors, and so is an immunosuppressive microenvironment. As you can find, very little activated T cells in these tumors, and fibroblasts have been shown to be at least one of the components that is involved in this process. So you can understand how, for all of these reasons, fibroblasts represent a promising target to develop new combination approaches for pancreatic cancer patients. And these are three of the main ways that laboratories and clinicians have tried to target fibroblasts. They have tried to reprogram them back to quiescence, to a state of quiescent fibroblasts. They have targeted specific pathways that are activated in CAFs, and they have targeted extracellular matrix components that the CAFs produce. So I'm going to talk about a few examples that are related to pancreatic tumors, but some of these drugs have actually been tested in other malignancies as well. Calcipotriol and Atra are two of these drugs that are used to reprogram cells back to quiescence and to remodel the microenvironment and the stroma. There have been promising preclinical studies with these drugs, which target the vitamin D and the vitamin A receptor respectively. And both of these drugs are currently in clinical trials to be tested for pancreatic cancer. Another of these drugs, but in the context of pathway-specific inhibitor, are CXCR4 inhibitors. This targets the CXCR4-CXCL12 signaling pathway. And I should actually mention that both the ATRA study and the CXCR4 inhibitor studies were done in Cambridge and in collaboration with other centers. And the CXCR4 inhibitor is also has been tested in a phase 1 clinical trial, both for pancreatic cancer and for colon cancer, and its clinical evaluation is still ongoing. The most famous examples of targeting the stroma in pancreatic cancer are related to targeting the Hedgehog signaling pathways by using SMUDin inhibitors. SMUDin is a component of this pathway, and Hedgehog signaling has been shown to be activated in CAFs in pancreatic tumors. These were very promising. There were promising preclinical data also came out from Cambridge actually in 2009 that showed that targeting Hedgehog signaling pathway leads to a downregulation of the extracellular matrix, which leads consequentially to an increase in the drug within the tumor and effective treatment. So due to these, following these studies, a number of clinical trials started with various SMUDin inhibitors, but unfortunately these trials were stopped because it actually turned out that either the patients had no benefit or had worse prognosis when they were receiving the Hedgehog inhibitor. Follow-up preclinical studies in 2014 suggested that prolonged inhibition of Hedgehog signaling compared to short-term inhibition of Hedgehog signaling, as the previous Science paper, was actually detrimental. And these studies concluded that targeting CAFs in pancreatic tumors is actually detrimental because CAFs have tumor restraining functions. Another very famous example of targeting the stroma in pancreatic tumor is PEG-PH20, which is hyaluronidase, which targets hyaluronan, which is one of the components of the extracellular matrix. Again, promising preclinical studies showed that targeting HA led to a decrease in the matrix and increase in drug delivery. PEG-PH20 was tested to phase 3, but unfortunately phase 3 clinical trial showed no benefit again for the patients that were receiving PEG-PH20 in combination with chemotherapy. It was interesting, this study, because this phase 3 clinical trial was actually one of the first that was biomarker driven. And so only high HA patients were enrolled in the trial and receiving PEG-PH20. And yet, unfortunately, this didn't show any benefit, and the PEG-PH20 will not develop further. So clearly, you can see that there is a disconnect between what we know about the function of CAFs, which have several tumor-promoting roles, and our ability to effectively target them. And so clearly, we need to better understand the biology of CAFs in pancreatic tumors and also in other malignancies. And so how do we study CAFs? Historically, the way that CAFs were studied was by using two-dimensional cultures. There's a number of reasons why this might not be the best way of studying CAFs. And the main reason is that the tension of the plastic of growing a fibroblast on a monolayer induces a number of signaling pathways like integrin signalings that induce a myofibroblastic phenotype in CAFs. But these are still helpful models to study some aspects of CAF biology. More recently, there has been increasing use of three-dimensional cultures, and again, various types of this type of cultures. You can use fibroblasts in co-culture with tumor organoids in matrigel. You can use them with hydrogels or with decellularized matrices. And during my postdoc, I co-developed one of these systems, and I'm going to talk a little bit about this in a couple of slides. But there's also a number of really underused models that I think that we should start thinking about to understand the biology of CAFs. And these models are organs on chips, where you can look at different matrices and combination of fibroblasts in co-cultures with different cell types. You can use air liquid interface cultures, which are short-term cultures that are kind of explants for tumors, and they are short-lived, but they give a better representation of a multicellular population. There has been increasing usage of three-dimensional bioprinted tissues, and just very recently, we see more lineage tracing mouse models that will tell us where these CAFs are coming from and how we can prevent fibroblasts or other cell types to become CAFs. One of the models that we use in the lab are the organoids. There's various reasons why we want to use organoids to understand the biology of CAFs. One of these reasons is that they grow in 3D like a tumor. And this might just seem like a fancy thing to say, but is actually biologically relevant, because if you grow cancer cells or fibroblasts on a monolayer, you are inducing their polarization to grow on the monolayer, whereas when the cancer cells and the fibroblasts goes in 3D, they maintain the polarization that they would have in the tumor. And this is related to a number of signaling pathways that is activated. Also, these models can be genetically perturbated rapidly and easily. Perhaps my students wouldn't agree, but you can imagine that once upon a time, to investigate what is the role of your gene in cancer progression, you would make your mouse model and observe how the loss of the gene is affecting cancer progression. Whereas now you can knock out your gene or overexpress your gene in the organoids. You can put back these organoids in the pancreas of a mouse and look at how that affects in vivo cancer progression. Importantly, also, the organoid models allow normal tumor comparison. And that's important because there's actually no normal untransformed pancreatic cell line that can grow in 2D. Whereas now with the organoids, we can grow normal cells in the same matrix and in the same media conditions than the tumors. And so we can then look at the differences, signaling pathways or therapy response that are really dependent on being a malignant cells or a normal cell. And also they recapitulate the genetics of the primary tissue. While 2D cultures can acquire mutations over time, organoids are stable cultures. And it's important because there is evidence that the genetics of the cancer cells is affecting the tumor microenvironment. However, the organoids are purely epithelial components, and this is great news for laboratories that are focused on cancer cells, because you can imagine that you can look at DNA sequencing, RNA sequencing, copy number variation analysis for your patient-derived organoids, and use the organoids as a tool to investigate the cancer signature in a way that looking at a piece of tissue wouldn't be possible, because you have so much of the stroma, as I showed previously, which could mask the signature of the cancer cells. However, for us that are interested in looking at the interaction between cancer cells and stromal cells, clearly organoids by themselves are not enough. And so for this reason, now a few years ago, we established a new three-dimensional co-culture model that allow us to co-culture pancreatic cancer organoids with pancreatic cancer cells, pancreatic stellate cells, or pancreatic fibroblasts in macrogel in growth factor-reduced media conditions. And these growth factor-reduced media conditions are important because they allow us to look at the symbiotic interaction between these two populations, with the organoids promoting the proliferation and morphological activation of the fibroblasts, and the fibroblasts vice versa, allowing the organoids to grow in these culture conditions. And so with these co-culture models, surprisingly, we found that actually fibroblasts are not homogeneous and are comprised of at least two different subtypes. A myofibroblastic MyCAF subtype, which is found adjacent to the cancer cells, and that has a transcriptional profile which allow us to speculate they are the cell population, the fibroblast population, that is predominantly responsible for the disposition of the extracellular matrix. And we found the second population, an ICAF or inflammatory CAF population, that is found more distally located from the cancer cells. And this is true in co-culture, but is also true in tissue in pancreatic tumors. We can talk about why ICAFs are more distally located and MyCAFs are more adjacent. If you're interested, you can ask me afterwards. So importantly, our discoveries that CAFs are heterogeneous is providing a potential explanation for these contrasting reports that we have about targeting CAFs. Some reports showing that they have tumor promoting function, and some reports suggesting that they may have tumor restraining functions. And so this shows how the tumor microenvironment and the fibroblasts are more complex than was previously thought, and we need to understand the biology of different fibroblast subtypes to know what functions or what population we should target for therapeutic benefit. And so we can use a variety of models to investigate CAF biology, not just the co-culture. We can use genetically engineered mouse models that have representative mutations from patients and that recapitulate the progression of the human disease. We can use tissues from patients and derive from patients and mouse models organoids, fibroblast lines, culture this organoid and fibroblast in co-culture, but also culture them with other cell populations such as immune cells. We can look at fibroblast heterogeneity by using a variety of techniques such as single-cell RNA sequencing, immunofluorescence, so RNA in situ hybridization, and specific markers for flow cytometry. And also we can put back these organoids and co-cultures in the pancreas of mice so that we can study again the mechanisms and heterogeneity of the fibroblasts overall to identify new therapies. And so with all these approaches, we have made new discoveries about the biology of fibroblasts in pancreatic cancer. We have found, as I mentioned, and confirmed in vivo that MyCAFs and ICAFs are the most abundant states of fibroblasts in pancreatic tumors, and they can interconvert, meaning that they are not endpoint in differentiation, but rather are affected by cancer cues to become a MyCAF or ICAF, depending on their location in the tumor. We also found a third population of CAFs, a smaller population called antigen-presenting CAFs, which might have a different cell of origin than MyCAFs and ICAFs. We can't make up CAFs from pancreatic stellate cells in vitro, for example, and APCAFs share several features with mesothelial cells. So perhaps mesothelial cells are the cell of origin of APCAFs. We also found the main drivers of ICAF and MyCAF formation, and this is important because it gives us some new signaling pathways to investigate and target to understand what is the function of these different fibroblast subtypes. We also know that ICAFs are involved in the recruitment of neutrophils and in the establishment of an immunosuppressive microenvironment in a particular subtype, molecular subtype of pancreatic cancer, so-called squamous subtype. And also as a note, ICAFs are the predominant producer of CXCL12 in pancreatic tumors. And CXCL12 is involved in this CXCL12, CXCR4 pathway that is currently targeted in preclinical and clinical studies. We also found new pathways that are involved in the maintenance of MICAFs, and I want to highlight the hedgehog signaling pathways. And we found that this is involved in MICAF and it's more highly upregulated in MICAFs compared to ICAFs. And this is important because, as I mentioned, preclinical and clinical studies targeting the hedgehog pathway were utilized to target CAF overall, because at the time there wasn't still the understanding of these layers of complexity of fibroblast heterogeneity. But what we found actually is that targeting the hedgehog signaling pathways with this LDE225 inhibitor doesn't affect CAFs equally. It predominantly affects the MICAF population, which is the one that has higher hedgehog signaling. And it actually leads to an increase in the percentage of ICAFs in the overall tumor, which leads to an increase in the ICAF-MICAF ratio. So you can imagine that while previous studies thought that targeting hedgehog signaling pathways was detrimental to the patients because there was a loss of a tumor restraining population, you can think that perhaps it wasn't the loss of tumor restraining populations, but the acquisition and enrichment of a potentially tumor promoting and immunosuppressive population, which caused these detrimental results. And so again, this is an example that shows how we need to better understand the biology of fibroblasts, and we need to better understand how our drugs are affecting the tumor microenvironment, because we might not be able to design new combination therapies if we don't have this complete understanding. So now I talked about pancreatic cancer and fibroblasts and why these are important, but you probably don't study pancreatic cancer, and so you may think, but why that is important for me? Well, actually, it turns out that fibroblast heterogeneity is not restricted to pancreatic cancer, but is a feature of several solid malignancies. And actually, the same inflammatory and myofibroblastic states that we find in pancreatic cancer are present in other malignancies. And the same IL-1 signaling and TGF-beta signaling pathways are present in other malignancies and in other fibroblasts, showing that looking at pancreatic cancer biology and the pancreatic cancer microenvironment can be used as a paradigm to understand, in some cases at least, the biology of other cancer types, and that fibroblast heterogeneity should be considered in other contexts as well. And also, single-cell RNA sequencing studies have shown that not only fibroblast CAFs are similar across different malignancies, but they also share the features with fibroblasts in inflammatory states. And so this adds an additional layer of complexity, because we need to be able to discriminate what is a feature that is typical of a CAF in a malignant state, or whether this is something that is also shared in an inflammatory state. And this is particularly important if you want to understand the biology of CAFs to design new diagnostic tools. And so there is a number of questions that remain to be answered in the sphere of CAF biology. One of these questions is, what is the cell of origin of CAF subtypes? I have showed that a number of studies suggest that many cell types, I would say all cell types, can acquire CAF features. But now we need to understand whether different cell types are originating different CAF subtypes. We know, for example, pancreatic stellate cells can be reprogrammed in both ICAFs and MICAFs, but not into APCAFs. And so lineage tracing studies, very lineage tracing studies, are trying to address these questions. And a few studies have looked at specific cell populations and how, what is their role in pancreatic tumors. Another question is that of the universality of CAF subtypes. I mentioned that there are some shared features. between fibroblasts in pancreatic tumors and in other malignancies. And very recently, there was a nice paper that showed that there are at least two universal populations of fibroblasts that are shared not only across different malignancies, but also across different disease states. And this is important because understanding the biology in pancreatic tumors might highlight some features in a different cancer types, and vice versa, understanding CAF biology in breast cancer may give us some hints of what's happening in pancreatic tumors. It's also important to understand how and what defines differences between CAFs or inflammatory fibroblasts or normal fibroblasts, so that we can design diagnostic tools, but also therapies that target tumor-promoting CAF populations without affecting other fibroblast populations. And we also we are, at least my lab is interested in understanding what are the bases of CAF plasticity and what determines this transition between, for example, inflammatory CAFs and myofibroblastic CAFs. And that's because this is a technical issue to study fibroblasts, because you can imagine that trying to, if you target one population A, and you end up with more population B, it's going to be really hard to interpret whether the phenotype you're observing is due to the loss of CAF A compared to the enrichment in CAF B. And this is what we see with the hedgehog signaling story, which led to an enrichment of one population and depletion of the other one. And so we are interested in dissecting this and also in developing new tools that will allow us to address these questions. And then, of course, we still need to understand what are the functions of the CAFs. And this is key because we need to develop combination therapies that are effective rather than detrimental. We need to define what are the populations that we should target, or perhaps features of population that we should target. And there's a lot of speculation about the function of these CAF subtypes based on their transcriptional profile or based on pharmacological studies that target signaling pathways involved in their formation. But we don't know how CAFs are affecting the cancer cells and how they affect different immune cell populations. And then finally, we need to still be able to translate this knowledge into the development of effective therapeutic strategies, keeping in mind that fibroblasts are one of the population of the tumor microenvironment, but treatments that might target fibroblast tumor-promoting fibroblast population and cancer cells may detrimentally affect immune cells. And so obviously the discussion is more complex than the one I gave today.